Okay guys, welcome back. I hope everyone's doing well today. Let's carry on with Unit 4, and we were talking about microorganisms and disease, and today we're going to start off talking about parasitic agents. Now remember, when we're talking about parasites, we're talking about or a parasitic relationship, we're talking about two organisms living together. We're going to have one organism, the larger one, that's what we call the host, and that's generally that's the organism that usually gets damaged. And then we have the parasite, of course, which is a smaller one, which causes the damage. So we break down parasites into three categories. We have the protozoans, and in the protozoans we looked at a couple in lab already. We looked at Cryptosporidium, and we also looked at Giardia lamblia. If you also think back to the first class, I talked about an amoeba called Naegleria fowleri, and this was one that was nicknamed in the press the brain-eating amoeba, but that's also an example of a protozoan parasite. We also have a group of animals which are worms, and we call those the helminths. And when we're talking about worms, we're talking about blood flukes or hookworms. We can also be talking about tapeworms or ascaris, the roundworms like we looked at in lab. And then the third group are our arthropods. These are going to be, ticks, fleas, lice. In the lab, we looked at bed bugs. We also looked at bed bugs, right? And the most common, of course, parasitic arthropod is going to be the mosquito. Okay, now this leads us to a couple of very, very important terms for you guys to know. Pathogenicity is the first one. So pathogenicity is the ability of a pathogen to cause disease in an organism. So what that means is, how easy is it for that particular pathogen to cause disease? Infectious agents have different levels of pathogenicity. Some, for example, are very highly pathogenic. It's very easy for them to cause disease in humans. So examples for that are Yersinia pestis, Listeria, and Clostridium tetani. Now, if you're not familiar, Yersinia pestis causes the plague. Listeria is a type of food poisoning, and Clostridium tetani is the reason that if you cut yourself on something dirty out in nature, you need to go to the emergency room and you get a shot in your butt, your tetanus shot. So these are organisms that can cause disease really, really easily. If you come in contact with them, there's a very good chance that you'll become infected and end up sick. Then we have, on the other side of the coin, we have organisms that have very low pathogenicity. So a good example of that is Staphylococcus aureus. We worked with it in lab. It lives happily on our body all the time, but it doesn't cause disease very often at all. It has to get into a situation, an opportunistic situation, in order for it to cause us any damage. So the level of pathogenicity, a high pathogenic organism easily causes disease. A low pathogenic organism takes a lot for it to cause disease. Now, the next really important term for you guys to know is virulence, or some people may call it virulence. Virulence is how severe an illness is. So if we have a pathogen that has high virulence, it can cause us a severe illness. If it has low virulence, then it causes a less severe illness. The two words together, pathogenicity and virulence, are often mixed up, so try not to get mixed up with them. Pathogenicity means the ability to cause disease, and virulence means how bad that disease is. So try and keep those separate and clear in your mind. Now here's a look at virulence again. Pathogens can cause disease. That's the definition of a pathogen, right? So pathogen with high virulence causes a severe illness. Pathogens with low virulence causes a less severe illness. So if we take influenza, for example, the influenza virus, a common influenza virus that you may catch almost every year if you're very unlucky, but if you get the flu, common symptoms for influenza are fever, cough, sore throat, runny nose, and generally it takes about a week or so to go away. If we look at another version of influenza, H5N1, this causes all of those symptoms again, but it may contribute to pulmonary failure, so your inability to breathe. It could lead to pneumonia. It could lead to multi-organ failure. So even though they're similar pathogens, the H5N1 virus is much more virulent than the common influenza virus. If we wanted to compare pathogenicity and virulence right now, we could look at SARS-CoV and SARS-CoV-2. Now the first one is what caused the SARS outbreak that you guys were just doing the lab about. This one down here causes COVID-19. Now SARS-CoV-2 seems to be much more pathogenic. It's very easy for that to cause disease. Once it spreads to you, it seems to cause disease in a lot of people, which is why we're seeing 300,000 cases, I think, worldwide now. So it's a very easily transmitted disease. SARS-CoV, the first virus that was we released back, or we found out in 2003, this seemed to take a lot more exposure in order to catch it. So SARS-CoV was less pathogenic. However, SARS-CoV had a much higher death rate, which would have made it a higher virulent. While SARS-CoV-2 doesn't seem to have that high death rate, even though it's causing pretty severe illness in a lot of people, SARS-CoV-1 appears to be more virulent than SARS-CoV-2. So these are just ways that we can use to compare these two different viruses. So pathogenicity and virulence, try not to get those mixed up. The next thing we need to consider talking about pathogens is a source of infection. Where do these pathogens come from? And we need to be very aware of the idea of a reservoir. And what a reservoir is a place where the pathogen can survive until it's transmitted to another host. A reservoir can be a living or a non-living object. And reservoirs can be a lot of different things. People can be reservoirs, for example, and how they're transmitted. If we look at the common cold, it's very easily transmitted by hugging someone, casual contact. You can also have transmissions of diseases or sources of infection from bodily fluids, blood, semen, vaginal fluids, saliva, tears. These things can all be transmitted from a reservoir, from a place where the bacteria or the pathogen lives, to a host through all of these types of things and interactions. Now, humans can definitely act as a reservoir. So we can have an infection in us and it can live there with us. Sometimes it'll make us sick and cause disease. Other times it may not. And then that virus or pathogen can be transmitted to somebody else. So if you have symptoms of a virus or a pathogen, you're a symptomatic carrier. But you could also carry this and pass this on to other people if you don't have any symptoms. You may not even know you have the pathogen, and we know those as asymptomatic carriers. Some examples of this include typhoid fever, and there's a story we'll talk about in a couple of seconds about how people transmitted typhoid fever through infected food. Pneumonia, which is at our forefront right now, can be viral or bacterial. Of course, we're worried about the viral version right now with COVID-19. Polio can be spread by an immunized individual. AIDS can be spread by a person. You can have AIDS and have no visible sign of infection, not know that you're sick, and still spread the disease. Now, we often class people as carriers. And a carrier means that this person has the infection and it's possible to transmit it to somebody else. So we have a bunch of different types of carriers: we have passive, incubatory, convalescent, and active. These things all represent different stages of infections in a person's life cycle, I guess, or stage of life. And it represents how they carry the pathogen and how they may transmit it. So if you're a passive carrier, for example, you have the pathogen, you can pass it off to somebody else, you can make them sick, but you never show outward signs of disease. So even though you have it, it doesn't cause disease in you for some reason. An incubatory carrier is somebody who has the pathogen, it's developing, it's beginning infection, and even though you haven't developed symptoms yet, you can still pass it to other people. So the measles, the mumps, polio, these are all things that are passed through an incubatory period. A convalescent carrier is when you've had the disease, but you're feeling better now. So you're convalescing, you're getting better, you're starting to feel a bit better. The disease in you has lessened. But even though you're feeling better, you can still transmit that disease. So cholera, typhoid fever, diphtheria, these can all be passed through a convalescent carrier. And finally, you can have an active carrier. This person has recovered from an illness, doesn't show any type signs of disease, but they can still transmit, and this can happen indefinitely. It can go on for long, long periods of time. Now, one of the great stories in the past is about a lady named Mary Mallon, and they called her Typhoid Mary. And the reason they called her Typhoid Mary is because she was a passive carrier of typhoid. And she didn't know this. She was never sick. And she didn't believe that she was a carrier. But she came from Ireland to the New York area in the United States. And she would work as a cook or a cleaner in rich people's homes. And she would pass typhoid on to the people who lived there. And she has been known to have spread the disease to, I think it was over 50 people. 3 of them deaths have been directly traced to her. But when the family she would work for would get sick, she would move and start cooking for another family. And this happened time and time again. The authorities would catch her and they would quarantine her. But then eventually she would get out and she would do the same thing again. Eventually she ended up being quarantined for the last, I think, 20 years of her life. And she ended up dying alone. But her whole life she was able to spread typhoid, and she became known as Typhoid Mary. Quite a famous case. The world's most famous asymptomatic carrier. Now, not only can people be reservoirs, we can also have animals being reservoirs. And when we get transmission from an animal to a human, we call that disease a zoonosis. And right now, of course, COVID is an animal reservoir virus. We've traced it back to bats and pangolins. And these bats and pangolins will have this virus, and if it gets transferred from those to a human, then we can have another outbreak. We have a lot of different organisms, or a lot of different animals that can act as reservoirs. For example, monkeys can act as a reservoir for malaria. Cats, your cat that you have at home as a pet, that can be a reservoir for toxoplasmosis or rabies. And that's quite a common virus that can be passed back and forth through cats. Dogs can also act as reservoirs for roundworms, toxocariasis, for ringworm, and for rabies as well. And then we don't necessarily need to be living in order to be a reservoir. We can also have non-living reservoirs. So soil, water, air, all physical parts of the environment but not living, they can all be a reservoir for infection. If you cut yourself, like I said earlier in the lesson, if you cut yourself outside, you can get Clostridium tetanus, or tetani, sorry. So that's a pathogen carried in the soil. Water can carry stuff like cholera. Anthrax can have airborne stuff such as influenza, measles, and tuberculosis.